We're almost to the end of the documentation. There's more to be done after the documentation, and I am stealing from another, again, I'm trying to steal from Titus's, but it's not quite working yet. So we're going to get through this stuff, and then we're going to take a pause, and we will talk, and then those of you who want to stick around, I will make it work, and you can see the last bits. That being said, we left off running the top hat, and then after that finishes, we need to actually get the counts. This again should look really simple, right? The only difference between what we did yesterday and what we're doing today is that it's A, a chicken, and the name is a little bit different, and B, instead of using the reference GTF, we're using the nostrand.gtf that we just made. So we can go ahead and copy and paste that in and have it run. And those input samples are again ready for EdgeR, except we don't have anything to compare them to yet. Right? Right. So, green stickies when you're ready to move forward, or in fact, I'm going to go ahead and scroll down. I've got this part running already, the next sample 2. I would say go ahead and move forward trying to get the sample2.bash script that I have on the web page running, and once it's done, stick up a green sticky and we'll move forward. And in the meantime, I'm going to play with making sample 3 on the screen. Okay, so now we're going to play a little bit because I don't have the files up and ready for you, and I apologize for that. We are going to change things. What I'm going to do is I'm going to make a new version of the sample2 file, and I'm going to save it as sample 3 to actually be able to run sample 3 rather than copy and paste. So for sample 3, I'm going to call sample 3 male replicate 1. And again, I'm going to change all of the bits and pieces from female replicate 2 to male replicate 1. There are lots of ways to do this using fancy copy and replaced. I'm not going to. Anybody see any errors that I made changing things? So once I change something in Nano, I can hit Control X. It asks me, do I want to save? Yes, I do. Oh. Where? Oh. Yeah, so this is going to fail for all of you. Just delete it. I apologize. That was a copy and paste from an old version of instructions. Okay, so in Nano, this is the two-step process, right? This is why it asks you two things. First, do you want to save your changes? Yes. What name of file do you want to write? Well, now it's a different file, so I'm going to save it as something different. And then it's going to double check, did you really mean to save it as something else? Yes, I did. So now, if I type ls, I have a new file, sample 3, that I can run. So I'm going to challenge all of you to make sample 3 as a file, and then also make sample 4 as a file, replacing replicate 1 with replicate 2. So I'm going to talk about the next part while we're running, just you can listen and think and tell me why I'm crazy. So at the end of this, we're going to be able to run EdgeR and get a list of differentially expressed genes. Right? That's at least our hope, because we're going to have 2 conditions, 2 samples each. We'll at least be able to get fake error bars and tell ourselves that there are some interesting genes that might be differentially regulated. The problem, and this was mentioned earlier when we were talking about what the annotation transfer did, or what the cuff merge did, is that it's going to use the gene names from the merged GTF, which have no real meaning, right? They are, in fact, not real genes yet. Those are the cuff-links created gene names. So the last thing that you'd have to do before you can get any useful information out of this is get the annotations from the genes that were real, or that were originally in the Gallus-Gallus reference genome. That part I can show you what we can do with today in terms of we can transfer those annotations over and get some useful bits. The part that's going to be a little bit more difficult to show today is what you do with the new annotations, genes that didn't exist already that you need to figure out what they are before you can get any useful information. The biggest statement in that is that there are lots of ways you can annotate the genes. You can do blasts, you can actually feed it through annotation pipelines. The problem is going to be pathways. If you actually care about what pathways those genes are in, there isn't necessarily going to be a way to fit them into currently known gene networks because they're going to be novel to the world so far. So we can talk about that some. I'm going to try to take you through the tutorial. This is where it's going to get a little hit and miss until lunchtime to try to get through that, and then we'll eat some lunch. You guys can go away if you want, and you can stick around and ask questions and play and see what else we can do after lunch. So there isn't an easy way to, at least that I know of, to just tell it to, oh, by the way, take the annotations from old and transfer them into new when possible. So we're going to have to do a little bit of fancy footwork, which fortunately there is a script for. So that part we can actually do. We're going to, again, pull from Titus's course because I haven't gotten it copied in yet because I didn't have a chance to test it last night. But those will transfer over to give you annotations that are deposited ensemble annotations that you can use in, we're going to use David today instead of PandaDB to show what you can get. Okay, since I'm still running, I'm going to work ahead a little bit by making a fake directory and doing things. So, okay. So I will show you where all these scripts are so you can copy and paste them later, but I have grabbed, because again, I know what you guys are supposed to do, so we have the data already available. I grabbed copies of the count files that we should be making right now, and also an R script that is going to read them in. I ran the R script, which took all of, here I'll run it again just to see how long it's going to take. 1, 1,000, 2, 1,000, 3, 1,000, 4 seconds. It will take somewhat longer when you have more counts, but this part will not change much. Again, R itself and doing a differential gene analysis is almost entirely dependent on the number of genes, not the number of counts in those genes. So all of this is going to take about 5 seconds to run every time you do it. Again, point here is that you can do all of that other work in batches and let it go and, set it, forget it, come back later. For the actual R analysis, the differential gene analysis, which is the last thing that gives you your spreadsheet that you want, do it by hand. Don't try to put it into a fancy script and wait for it and everything else. It's not going to take long enough to wait for. So do the R by hand because you're going to want to be able to change things and check things and all of that. So we again made a CSV and a couple of plots. In this case, oops, that's not what I want to do. Let's try. It outputted 2 different plots, which is good. So I was asking about batch effects yesterday. What do you guys think about this? This is the plot that comes up when you ask whether or not there are batch effects.
PCA result?
The equivalent of, yes. It's what components it's creating is not actually individual genes. It's averaging several gene expression values, it's averaging the most differentially expressed genes, and I believe least differentially expressed genes is your X and Y axes here. So it's trying to show you what should be showing up. So, do you guys think that these are exhibiting batch effects or not? Is there such a thing as a batch effect in this case? Blastocysts, samples from individual growing embryonic chickens.
So female replicate one and male replicate one is on the same batch? Or what's the experiment design?
That's the question. No, these should all be individual samples.
Okay.
So is there a possibility of batch effect here? Well, yes, insofar as all of the metadata that are not available might give you some information. So there's also, this is where you start getting into real problems about not having enough replicates. I can't tell you from this whether or not there are real batch effects or not, because I only have 2 samples of each, and they're equally as far away from each other as they are from opposite conditions. There could be a batching going on, there could not be a batching going on. Were all of these eggs in the same incubator? Were all of these eggs from the same hen? Were all of these, the list goes on and on and on and on and on as to what could have caused separation between these. This MDS plot is vitally important, however, when you have an adequate number of samples, especially if you're concerned about the possibility of batch effect. Batch effect doesn't always have to mean same sample or same individual. It could be something as simple as what day these samples were gathered on, or what time point. If you're doing a time series in bacteria and you extract at 15 minutes, but you do 2 separate extractions, it may be that day 1 was 15 minutes, and day 2 was 15 minutes and 12 seconds. And the gene expression is differential there because of that timing, and so you're going to end up with batches that you didn't expect. The secondary statement there is that I cannot stress enough how little respect metadata is paid and how much it makes me crazy. The more things you record about a sample before you take it, the better off you are. The likelihood is very high that you will never need to know who actually extracted the sample, or what time of day, or what color the egg was. However, you don't know until you get to this plot, which should be, at best, this is going to be about a month after you collected the data, because it has to get sequenced. You never know what this plot is going to look like, and if you need to find any factor that might be correlated between all of the samples that group together that shouldn't have, metadata is the only way to find it out. So record metadata, please. And by metadata, I mean metadata about an individual sample that is not related to the steps that you have to take to get from A to Z. Time of day, location of sample, anything that would be different between your sampling conditions is vital to problem solving. If your samples are perfect, then you'll have wasted time. If they're not, you will be very thankful. So just bear that in mind. Again, pet peeve of mine, I have seen it in almost every kind of project I've tried to work on, that there's some sort of data that I'd really like to have had that I didn't because it wasn't recorded anywhere. So just bear that in mind. So that pops up a great report. This is the MDS. I'll show you. Well, you saw yesterday how to deal with it, or how to generate it if you want, but we'll look at it again. And then here's the actual plot that we saw yesterday, eventually. Chickens. Much more difficult to deal with, especially because what's the difference between these two conditions is gender, and we only have 100,000 reads to work with. We get two dots that are red, which again means that there are 2 genes with an FDR above or below 0.1 for our sample. This would not be good enough for most people. Well, it might be, right? If this were real, if you had 50 million reads per sample, and you only got 2 genes, this is usually cause to celebrate, right? I have to do 2 follow-up qPCRs, and then I'm done, and I can publish this paper. It's usually not going to be the case. A lot of this is because of the variation and not enough not enough reads to actually give you good value.
So this is based on like the total read count, right?
It is, so reads counts per gene is on a log scale on the x-axis. Log fold change is the y-axis.
But like the different chickens, the total RNA you input may be different. You need to normalize the total read count too.
That's EdgeR's job, not yours.
What?
That's EdgeR's job. The tool itself, the R script itself, is going to do all that work.
Oh, they already done that.
That's all done by the tool itself.
Okay.
Don't try to do the work yourself, right? I hammered on this yesterday, and you missed it. But don't try to do fancy statistics unless you're getting a PhD in statistics. The tool exists. Use the tool that does the job right. If you need to understand what it did, that's great. EdgeR, like I said, has an 80-page manual that is really, really, really good, so use it. Oops, that's not going to recognize. Let me quit out of that. Okay, so we did our R scripting, and now, is there a question? Or are we just seeing all the other things scrolling up? So now we have our genes, right? Everything else has an FDR of 1. So the likelihood that that's a real gene is essentially 50% that we actually found it. It's a coin flip for each one of those as to whether or not it's actually differentially expressed. We've got a couple at 0.9, and then here are our 3 genes below 0.2 in this set, right? But again, the problem here is that XLOC is chromosome location gene number, and that's it. It's not the actual ensemble or NCBI gene annotation. So what do we do now? All we have is a list of genes that are completely useless. There are a lot of different ways to transfer the annotation. However, I'm going to use this. There's a link at the bottom which links to one of the pages in the old tutorial. You can kind of back up and find this, or I will put up a better link in a minute. So what did I do, right? This was a script that, again, doesn't get rid of the old information, but all it's going to do, and it's written in Python, if anybody wants to learn how to write scripts in Python, we teach a class on that too. It read in every one of those and added in information about the gene name and the annotation ID between the original locus ID and the fold differential change. That's all it did. Easy script. But now what we have is a list of genes that are actually something that we could be interested in. So I'm going to take these top 5. Oops. And again, I'm going to paste them into Excel. And do fancy text 2 columns. Next. Finish. And say, well, this is interesting now, right? So of those 4 genes that I copied, 2 were novel, or essentially novel, right? They didn't have a previous gene ID associated with them, and 2 have those gene names. So what can we do with that information? Well, we can look up the genes individually. We can go to Ensemble or NCBI and look up that gene ID and see what gene it's associated with. We can look at David. Oops. See if this actually works. So David is another annotation database, or can't read very well. There we go. Oh, chose the wrong thing. I'm going to take a pause and just talk about it. So David is, as it's listed, it's a gene ID conversion tool. It is taking your gene ID and giving you useful information about it. So if we wanted to, NMs are, no. Let's see if we can figure it out. So all of this is well and good, and again, we're using fake data because we've only got 1,000, so we can take more of the genes and try to fit them through. But at the end of the day, no matter what we do, we're going to be able to use this information to get a list of genes that we already have, or genes that were already annotated before, to see what might be differentially expressed according to that bit and piece of information. See if David can handle that. Can't. So what we're going to do then, gene bank. So of those 2 genes, we can see what functional categories they are falling under. We can look at the gene ontology. Try to see all that stuff. Now with 2 genes, it's going to be really hard to see anything because all we really have is 2 genes. So instead, we're going to cheat, or maybe not cheat is the wrong word, but we are going to use more genes than are actually differentially expressed. Maybe. If the file will actually show up. So at the end of, when we get there, we will do it, and I will talk about it in a minute. The other thing that I did want to talk about is what we can do with the rest of them. So there are a bunch of different tools that you can do things with. The suggested protocol here. So let's just assume for a moment that we got all the way through David with 1,000 genes and got some really interesting information about the annotated genes and what we could possibly know about the difference between male and female development in chickens based on that, but we still have a bunch of unannotated genes. So what can we do? So when you're saying that, when you're asking about that error from the FASTA file, we're going to have to go back if we want to do this and regenerate that FASTA file now because we're interested in the transcripts that didn't exist before. So to do that, what we would do is we would use something, so in this case we're going to use transdecoder to take all of the annotated genes that we have predicted out of cuff links and apply those to transdecoder. What is that going to do? That's going to take your gene sequence and turn it into a protein sequence. Again, outputs are difficult, so there's another script here to get rid of stop codons and then get a new set of peptides. Once you have peptides, what can you do with them? Well, this is what it tells you, right? So this would be the output, is that we have some sort of transcript, and it's a complete transcript, and that's all we know. Here's the protein sequence. Okay, we're done. We can all go home. We know that it's protein. I mean, if you want to know what it does, you can try other tools like in a ProScan. However, it's not quite working yet, or at least it wasn't a year ago. I'm not sure that I've gotten InterProScan up and running as of lately. So it's probably still broken. However, it will give you output if you run it, and there are lots of different ways to do it. That looks a little bit more useful. Right, so InterProScan is a tool that we should have installed if we don't already. So if you're going to be doing this, feel free to ask us to help you. It takes protein peptide sequences. Here's the name. Here's some fancy text. Here's how it decided what it got, and here's what it thinks it does. So now you've got a set of a whole bunch of the unannotated genes, and you know that some of them appear to be immunoglobulins, some of them appear to be part of weird gene superfamilies. You've got genes and genes and genes. IG, a lot of immunoglobulins, a lot of kinases, et cetera, et cetera. So again, once you have that, we're into you can't do it all on the computer. All of this stuff, be it generating of the CSV file that you are actually going to use because it has a differential gene expression, be it taking any useful gene IDs and taking them to David or Patrick or PantherDB or any other tool set of given gene annotation, give me gene pathway kind of stuff, is all stuff that you're doing by hand, and there's going to be a certain amount of custom work involved in any of it. So in this case, even if you run InterProScan, it works great. You're still going to have a list of all of your peptides from all of your genes, and you're going to have to decide which ones of those are actually differentially expressed and try to combine that information with information from your InterProScan output and make a brand new analysis based on which genes you think are interesting, which genes might actually be useful. And all of that is going to be dependent on what your project is, what your time frame is, how much effort you want to apply to finding out what a new protein does versus working on proteins that already exist because you don't have time to do molecular toolkits and change proteins and all of that work if that's where you're at. So all of that, as I said, is ways to get quote-unquote better annotations, different annotations out of differential gene expression that came from a semi-model organism. It gets a little bit harder, right? Yesterday it was pretty straightforward. The gene, you ran the alignment, you got an answer of which genes are differentially expressed, you pulled that list out and annotated it, looked at what those genes might be, and were able to move on with your life and say, these are the genes that I'm really interested in, so maybe I can do some follow-up work. Now we have a semi-model organism. Some of those genes had annotations that were differentially expressed. We could relatively easily go through, look at what those genes were, what they did, see whether that made sense, whether that was interesting, something we wanted to follow up on. But now you have all of these extra proteins that we don't know exactly what they do, but we're going to use a couple of tools to take guesses and hope that that was the right annotation for those tools. And if any of these are interesting, even though the original stuff that was annotated isn't, I have to go through a bunch more steps to validate that these are real, that they're genes that need to be annotated. When I publish my paper, I'm going to have to go in and put all of these genes in to be added to the genes.ggf file for chickens, et cetera, et cetera. Now imagine for a moment that you're working with an organism with no gene models, no annotation, and all you're going to be doing is this work. You're not going to have time in the lab. You're going to be doing this for a year, probably, right? These are all of the things, welcome to bioinformatics, I'm sorry. The farther down the road of non-model you get, the more and more work you're going to be doing on the computer, the more and more work you're going to be doing with tools that are either not perfect or not invented yet. There's my doom and gloom statement for bioinformatics. The other side of that is that it is a really awesome field. There's a lot of really cool things you can do because you know how to use computers, you know how to use these tools, you know what the limitations of these tools are, and you know what needs to happen next. It is an exploding field. There's a lot to learn. It is really difficult. It is probably not worth everyone's time, but it was certainly worth mine. So I won't tell you to run away screaming, but I'm going to at least give you a healthy dose of warning that you're going to spend, if you want to really do bioinformatics, if this is a career you want to go into rather than just getting your answers and going back to the lab and validating them, you're going to be spending most of your time in this world, not in the world of yesterday. So, model organisms, easy. Semi-model organisms, a little bit harder. No reference genome, no annotation, a little bit harder. Metagenomes, even harder. Alien biology. I guess if you know somebody, feel free to get into it. Sign me up too. So, I apologize for this last portion being a little bit nebulous. You see the link up here. There's a read the docs link. You can start at that if you want to see what we taught a year ago and try to use that for any bits and pieces that I may have gotten wrong. There's a lot of stuff in there in terms of the copy and paste that I would not recommend copying and pasting when it involves the command line. I tried a lot of the stuff that moved over with these new examples is to make everything a little bit cleaner in your home directory so that everything stays in your example directory, everything runs appropriately, and all the data are available and don't end up wasting lots of extra time. So, I'll stop there. We've got about 10 minutes. We can ask questions, and then I will show you, hopefully I'll be able to show you what real data look like going in for the human sample from yesterday. So, I skimmed over a bunch at the end here, but we also talked about a lot of stuff. So, questions, thoughts, ideas of how to make this apply to your own research, now would be a good time.
So, like you mentioned before, using Top Hat, like if you're dealing with bacteria, wouldn't be a good idea. It would be the best route to go. So what are some other tools that you would suggest using?
So the reason why Top Hat is a good tool for eukaryotes is because it is much better at handling the gapping that occurs in RNA-seq with introns and exons. If you're trying to deal with something much simpler like a bacteria where you don't have that, or if in another case, the other place where you would do this is if what you have is a cDNA library and you don't have a reference genome, the cDNA is the message, so there's never going to be gapping in that transcript, right? It's going to be the whole thing that you're aligning to. In those cases, you can use any read aligner, right? What you need to get, let me describe that a little bit better because that is in back to the pipeline. So an RNA-seq pipeline is always going to be QA/QC, get the right reference genome, align the reads to the reference. Once you do the aligning of the reads to the reference, that generates a BAM or a SAM file. Once you have a BAM or a SAM file, this portion is all going to be the same. It's still going to use HTC to take that BAM file, turn it into read counts, and then you can do whichever pathway it is, depending on how well your genes are annotated. Using top hat here is useful when the genes are going to gap for one reason or another. Using something like BWA or bowtie 2 is a perfectly legitimate alternative to that if you're dealing with a reference genome that does not have introns or exons, either because it's a bacterium or because your reference genome is in fact just a cDNA library. So that's the point at which something is different because of your reference, right? And those two kind of go together. Tool selection and reference genome selection are probably tightly coupled. Again, the counting of reads and doing the differential gene expression is centered around using EdgeR or DE-seq as your analysis tool. There are a lot of papers that you can go and find and a lot of arguments that you can go and read of why that is a better or not better way than using a method like the cufflinks, cuffdiff, cumberbund mechanism, which uses RPKM values instead of absolute read counts. To take a minute to describe that, the difference is absolute read counts here is just counting how many reads aligned to any given gene. It does not care about the length of the gene. It doesn't care about the number of total genes in your sample. When you do your analysis at the end of the day, it's going to care about the total number of reads and how many reads align to any given gene. When people were doing a lot of this at the beginning, there was kind of a split between that and people who believed that the length of the gene needed to be taken into account when you did quantification and differential gene expression. That led to a whole bunch of questions and ways to deal with it, and the most important of those is the idea of the RPKM value, which is reads per kilobase per million reads, which is for every million reads you had, how many reads aligned to a given gene normalized to its length. So it's essentially normalizing all of the gene expression values to the length of the gene. If you want to do comparisons of gene expression of a single sample, so if you're just doing differential gene expression between multiple samples, that has absolutely zero value. If you're interested, however, in comparing gene A and gene C within the same sample, RPKM becomes much more valuable because now you can actually do a straight one-to-one comparison. If you use the count reads that we generate here to do that kind of comparison, I won't say that you'll have a bad time, but there's certainly a potential for several orders of magnitude of error to enter into that equation because of the length of the gene. That's where you might actually want to go back and look at the top hat cufflinks cumberbund path instead. Yes. Any other questions? I'll stop at that with that statement, but anything else? Okay, so we've got half an hour before food gets here. We can talk about lots of things. Do you want to try to do the analysis on the data from yesterday? Do we want to do something else? What would be the most entertaining and useful for the people in the room? Suggestions, or should I do show of hands? So the two options I can think of off the top of my head are to try to poke the David database a little bit more firmly and show you what it looks like, or to go back and show you what differential gene expression looks like with a full set of reads instead of 100,000. So option A, David. Option B, model organism RNA-seq with a full set of reads. So vote of hands, option A. Database, David, option A. Option B, completely apathetic and don't care, just staying around for lunch. Okay, yeah. Okay, so let's take a look then at the B's habits. So let's look at reads. Is this big enough for everybody to see other than the fact that the people in the back can't see the bottom? I can make the text bigger. I can't make my screen stretch to the top. It's dumb. So I'm going to just do a lot of clears here. But in the meantime, everybody should actually have this in their RNA-seq model. If they don't, I will show you all how to rerun it, but there should be a directory called full, and that is the full analysis because I couldn't think of a cleverer way to make that. So let me make sure it actually ran. I submitted it last night. Yay, accepted hit.bam and counts. So what I didn't do, because I am a terrible person, is actually go back and validate that each one of these names is what I think they are. So the first thing I'm going to do is do this the hard way. Just hold on that back. And we're going to go to Ensemble. We'll just search that way. That's really not helpful. Why is nothing giving me? There we go. We can tell this is the right link because it takes a minute to load. So I went into EBI and went to the expression atlas because this is where this experiment came from. And ERR 315325 is salivary. So oops, that's not going to work. Let's try that again. Long. So the other is salivary. So all I'm doing again is making my list. Now, I can do a lot of fancy things because I know Bash, and I'm going to do it now because I'm going to hopefully make sure that that's helpful, which is going to be... No, that's not going to work. I take that back because it will be several hours of error, so... Okay, so now all I did was copy those count files that I generated last night down here. So we have lung rep 1, rep 2, salivary rep 1, rep 2, right? See them? Everybody okay with that? So just to give you an idea, just as we had before, right? It's the same ensemble gene IDs. They're just way more counts. Right? Before it was 1s and 2s. Now it's 0s or 90s or more, which is a much better number to have. So So I copied over this file. Ruffle 1, 2, cancel, okay. I have to make one edit because I messed up the name. And there are fancier ways to do all of this, but I'm trying to hurry. Okay, so now I have made this directory almost an exact copy of the other one, so this should work fine, which means that it's not going to because I'm trying to show all of you this live. Well, that's a problem, isn't it? So we have a problem, which I'm going to ignore by pretending that there's only one lung output. Apparently my lung output didn't finish last night. So we are going to do 3 instead of 4. It was lung 2, right, Duranya? It was lung 2 that was empty? Where is that length? Oh. There we go. So 5 minutes of poking, and now you can see it's going to take a little bit longer, but it finished already. So don't care. It's a lot of stuff, right? Remember what I said yesterday, though. This is using an FDR of 0.1. So we got way too many. And again, this is just a graph, so you can go through and you can filter manually. But we can also change just the graphing requirement without doing anything else. Let's see what we get if we change this to 0.01, shall we? I haven't done it, so we're going to find out. Oops. That won't show me anything new. I have to actually do the analysis. Anytime you rerun something without changing output file names, it just rewrites it, which is, again, why you have to be careful when you do things like this. But I'm happy to break things because it's not my data. I'm not going to say that this will give you a lot more, but it should, or a lot fewer, but it should at least be slightly less demonstrating.
Is an FTR of 0.1 pretty standard?
No, 0.1 was because the data wasn't showing anything at, say, 0.01 or 0.05. So what are we seeing here, right? We're seeing exactly what you expect is that a lot of differential expression is going to lead to almost, in almost every case, if it's out here, it's going to be accepted as different, no matter which, right? All you're doing by changing it is just editing the edges of that curve. Part of the reason is because we only have one sample for one of those two, so there are no error bars on those counts, which means that any of these that might have high variance aren't going to show up unless or until we get the lung counts in there. The more samples you run, the more discrete this is going to be. You also have to bear in mind that this is healthy tissue, lung versus saliva. How many genes do you really expect to be differentially expressed between lung tissue and salivary gland tissue? Almost all of them, right? So it's not surprising that you see a chart like this for the experiment that you're looking at. But it does look much cleaner. The other thing that was pointed out to me yesterday that I think is kind of interesting is that the version of R also dictates the version of this plot. Most likely because there's a more recent version of edge R in there. So updating to a more recent version of R isn't going to change much, but it is going to look just a little bit different. And this is either because of the version of the software or the version of the edge R tool. So it actually didn't change much, but the lines are a little bit curvier. I can't tell you exactly what changed in the way that it's calculating things, but it did calculate things slightly differently. The numbers are still mostly the same, but it is actually kind of nice because this one gives us a nice example of something with a very extraordinarily low fold change that is inside of these, but is actually still red. And these are the ones that are outliers that would never be caught if you were going to do a simple fold change analysis. And may in fact be the same one that we were looking at before. From the smaller data set.